So I'm going to apologize up front. One of the things that I realized as we went through our journey yesterday was I prepared a wonderful presentation, but it was the wrong presentation for this audience. So I spent a little bit of time last night making some modifications. So if I go through a couple slides quickly, it's not because I'm having a senior moment up here and I've forgotten what I was going to say. It's there. I put some things in here that I really want to make sure that I have time to talk about, and frankly, 45 minutes goes by really, really quick when you're standing up here. All right, so I guess I promised in the first slide a trip down memory lane. We're going to remember what E&L used to look like before 665. Hopefully that'll be a justification of why we have 665. I'm going to spend a little bit more time than I thought I was going to spend on the document today, what it is, because I couldn't help thinking more than once yesterday that are they talking about the document that we wrote? Where are they getting this stuff? And I have to admit, I spent a fair amount of time yesterday looking through the document and making sure that it wasn't just me that didn't remember. If there's anything that I hope you remember after the 45 minutes together, is that the USP is your friend. The USP is your friend. In creating the E&L chapters, we really strongly advocated for the industry perspective. And often that's also the regulatory perspective, but it's not always the case. And so I want you to understand that when we wrote this document, we wrote it from the perspective of people who would have to implement it. People would have to live with the consequences every day. It may not always be obvious, but that's at least the mindset that we had. And then as time permits, I'll look into the crystal ball and at least give you my opinion as to what happens on December 1st, 2026 and beyond, specifically with respect to USP's position on how to qualify manufacturing components. All right, going back to the real beginning, when I joined Baxter more years ago than I care to remember, this thing about manufacturing equipment just really didn't exist. For the first 10 years that I worked for Baxter, when we talked about manufacturing, it was a stainless steel mixed tank, stainless steel tubing filled in directly into a bag. There was no plastic in the process stream. So the issue of organic extractables and leachables didn't exist. And then all of a sudden, single use. You started hearing about single use, and at the very beginning it was, you'd occasionally hear about it. And then all of a sudden everyone was talking about single use, and single use became the way to go. A great concept. Cleaning validation is a real problem sometimes. Maintaining meant to last forever facilities is not a simple proposition. But I remember in the early times, when people would get up and talk about what are the impediments of implementing single use. And number one was extractables, and number two was leachables. Okay, the two biggest impediments to getting on board with the revolution was extractables and leachables. And it's not hard to understand. I mean, I don't know what your first impression was when you saw a manufacturing a system that was predominantly single-use technology, but I know what my impression was. Man, that's a lot of plastic. No wonder extractables and leachables is number one and number two on the list of concerns. There's a real possibility here that we might have issues. Now, interestingly enough, when we end this presentation, you're going to hear me say exactly the opposite. That extractables and leachables, the gut reaction that extractables and leachables was going to be a significant issue, turns out not to be the case at all for very good reasons. Okay. I still get this today, so I mean, I have a hard time saying this is what it was like in the past because it's still like this. When you go out there and you say to a client comes to me and says, "Hey, I want you to do a 665 risk assessment on my extractables and leachables information," and the first thing you ask is, "Okay, what extractables and leachables information do you have?" And I've got all sorts of stuff the vendors gave me, and all sorts of things I did in the laboratory. And then you look at it and you say to yourself, "My God, this is not extractables data. You gave me NVR in water extracts, or you gave me a bunch of compliance documents." And so the fact of the matter is that the importance of extractables and leachables to the implementation of single use was met by the fact that there was chaos out there. Ask two people to give you extractables information and you get completely different things. And sometimes you get the blank stare that said, "Extractables? What are extractables?" So that's the scenario that the USP found itself in. And frankly, our job was to bring order to the chaos. Okay, and we took that job very, very, very seriously. You kind of have to step back and put yourself in the position of the USP. We had written chapters for packaging at this point, and we found that the most difficult, or the biggest impediment to truly out of the box thinking was the fact that we walked into a space that was mature. Okay, every lab had their analytical methods, they had their preferred extraction techniques, and so on and so forth. And so the idea that somebody like the USP could come in and actually propose a standard extraction protocol for container closure systems was a dead on arrival. The cry from industry that no, we're not going to give up our own methods and so on and so forth would have been so great. But that wasn't true. That wasn't true for manufacturing systems. We were walking into chaos and we had the ability to do almost anything. And so we said, what the hell, why not? Why not? We're going to do what nobody else can do for packaging, and we're going to propose a standard extraction protocol. And people like me are going to get up and try to justify that standard extraction protocol. And we're going to talk about things like, what's the solvent, what's the time, what's the temperature, and we're going to, expect the pushback and deal with the pushback. Okay, so that, again, I'm trying to set the stage so that you understand what our mindset is, because if you understand what our mindset was going in, maybe it makes it easier to understand what the output was. Okay, this is really important. It's understanding the essential role of the USP. So we, as an organization, we can establish the means by which data is generated and interpreted. But as an independent organization, obviously we have no say as to who should perform the testing. Now, I have a personal opinion. in terms of who should perform the testing. But again, you need to understand that it's not USP's perspective to say whether it's the vendor, the user, or someone else who has to generate the data. USP just says it's the user, it's the sponsor, it's the person who is looking to market the drug product that has to have the data. I don't care how you got it, but you have to have the data. I guess the other thing to get across right away is the USP is not an enforcing organization. The FDA in the United States, the FDA enforces the regulations. The idea is, and sometimes the idea works really well, and sometimes the idea doesn't work very well, is the idea is that the USP sets the law and the FDA enforces the law. That's the idea. And so in that spirit, in addressing the manufacturing equipment space, the USP generated 2 chapters. A chapter below 1,000 and a chapter above 1,000. Now, people have talked a little bit about, well, one is enforceable and one is informational. And, I've got to tell you, the truth is, even the USP doesn't believe that. Let me give you a slightly different perspective as to what the difference is. The slightly different perspective is that a chapter below 1,000 is supposed to communicate tests, test methods, and acceptance criteria. That's it. Okay, these are the tests you have to do. These are how you do the tests, and these are the acceptance criteria. Okay, when USP does that for a simple monograph, that's perfectly adequate. But for a complicated monograph like an extractables and leachables monograph, a chapter below 1,000 that just tells you what you have to do and how you should do it and how to judge the results leaves a lot of questions, like why, or when, or how, or who, or what if, so on and so forth. And so with the advent of extractables and leachables chapters, the USP created this dual system where each chapter below 1,000 would have a mirror chapter above 1,000. And it's the mirror chapter above the 1,000 that you would go to and get the explanation. For example, why did they pick 50% ethanol? And why is it 40 degrees for 7 days or whatever? And I don't exactly fit into any of this pigeonholes that's in 665, and what am I supposed to do, and so on and so on and so forth. So it's created, again, it's not just 665, 663 and 664 to be written or already have their informational chapter. So it's a USP strategy moving forward. Funny things happen in the real world. And I'll use 1663 and 1664 as an example. The way I've just described them, those are meant to be informational chapters. They don't contain necessarily any requirements. Lo and behold, as you get FDA deficiency letters on container closure systems, you see the language, we suggest that you follow 1663 and 1664. And all of a sudden, chapters that we wrote to be informational are enforced to be mandatory. So I encourage you to get out of that mindset. Below 1,000 is mandatory, enforceable, and so on and so forth, and above 1,000 is just for information. It's whatever the regulators decide. Let's skip this. It went through the cycle 4 times. That's actually a record. It just goes to show you how hard it is to come to a consensus on some of these topics. I swear, every time we changed a sentence that industry liked, the regulatory agencies said we can't buy into that. And when we changed it to be a little bit more friendly to the regulators, the industry people would come up and say, you know what, this is way too much work. You're not my friend anymore. I'm not going to buy your books anymore, and I'm not going to read your articles. So it was a no-win. It was a no-win situation. It was trying to find a compromise between two viewpoints that are diametrically opposed. So again, I'm just going to apologize up front. If you don't like what's in 665, all I ask you to do is think about what it could have been. All right. This whole thing about a grace period, again, this is unique to E&L chapters. Before E&L chapters, there was no such thing as a grace period. The USP working groups came up with their monograph, and the minute that it was published, it was official. Okay? And again, what we say in public is, well, it gives people enough time to switch gears and be compliant on the day that it becomes official. But there is a behind the scenes a reason as well. It's a test drive. It's a test drive period for the USP. It gives people the opportunity to test drive our monographs, our chapters, and to give us feedback in terms of what doesn't work, what isn't working as well as we had planned. I really wish that I could say that the USP always got it right 100% of the time. I'd actually settle for 50% of the time nowadays. But I mean, again, the fact of the matter is when you're trying to establish ground-breaking guidance, you don't always get it right the first time. And I know we look at December 1st, 2026 as being the date that it becomes official. But I look at it as, man, we started writing this in 2015 based on the knowledge we had in 2015. And there's been an awful lot of knowledge and an awful lot of experience and an awful lot of published between 2015 and now that in retrospect makes us look at the monograph and say, well, if we knew then what we knew now, maybe the monograph wouldn't look that way. And so one of the things that I'm going to suggest at the end is 665 is already ready for a revision. It hardly seems right, but the USP has a process. It's an official process that every monograph is reviewed and revised as necessary on a 5-year scale. Well, if you start that scale at 2015, or even if you start that scale in 2021, where the third draft showed up in the Pharmacopoeia form, we're almost at that 5-year. We're almost in that 5-year point. So USP 665, almost on the day that it becomes official, is going to be up for revision and review by the USP. Okay, again, I don't think that I need to focus on that slide. Let's talk a little bit about this. In scope and out of scope, okay? It's interesting when you're sitting in the room and discussing what the monograph should look like, what kind of concessions you can get, and what kind of concessions you can't. And one of the major concessions that the writers of 665 got was scope. Originally, the scope was everything. Everything and anything. Everything and anything that has to do with manufacturing of pharmaceutical products should be in scope. Well, it didn't take too long to realize that's a pretty big scope. Okay, and where can we cut corners? Those of you that maybe remember the genesis of 665 when it was 661.3 might remember that there was language in there about material characterization. You characterize materials, and that's what tells you, what materials to use in your component. That's out of scope now. Okay, so the chapter became simpler. This may not sound like a very big concession, but if the component is in contact with a liquid process stream, it's in scope. If it's in contact with a non-liquid process stream, it's out of scope. And what out of scope means is not that we don't have an opinion. What out of scope means is the opinion that it doesn't need testing. Okay, and maybe you're scratching your head and you're saying, well, Dennis, what process stream is not liquid? Well, the most obvious one to me is a frozen drug product, a frozen drug substance, the storage of a drug substance or a drug product frozen. Okay? By other measures, maybe surface area to volume and duration of storage, you would think that a container for the storage of a drug substance would be high risk. Okay, it's in there for a long time. It has a large surface area. Drug substances may not be the simple inorganic or the simple buffer solution, that characterizes a low risk. But okay, so now all of a sudden a potentially high risk situation is completely out of scope. It's as close to no risk as we can get. The USP says if you store a drug substance frozen in a container, no extractables or leachables information is required, to qualify that container closure system, with a caveat. Okay, and the caveat is, well, of course, if you store it frozen, it has to be thawed. Okay, so the time that it's frozen, it's out of scope. The time that it's liquid is in scope. Okay, so it's a partial victory. Bullet point number 3 is a major victory, and frankly, something we were surprised that the FDA gave us. The FDA told us, as we were discussing scope, that they really weren't concerned about what they called APIs. And an API is just a drug substance. Again, I struggle with how to explain it. It's a traditional small molecule kind of a drug substance. Okay, they said, listen, anything that goes up to the production of this traditional small molecule drug substance, this API, is not of concern to the FDA because we think APIs are sufficiently well characterized that the risk of extractables or leachables in a traditional small molecule a drug substance is small. So it's out of scope. It's out of scope. I wish one day we could get there for biologics as well. But we've managed to do that for, again, APIs, traditional drug substances. What about these auxiliary items? Gosh, I got a lot of questions when USP 665 first came out about, what about scoops and volumetric flasks and, all these things that I use to transfer beakers and so on and so forth. Are you going to tell me I have to extract these with 50% ethanol and so on and so forth? And no, of course. Of course not. The answer to that is of course not. The duration of contact with those kinds of items are so short that it just doesn't make any sense that one would do any kind of extractables and leachables testing of these auxiliary items. So they're out of scope. And we already talked about the frozen storage. In the development of 665, if there was one thing that we repeated to ourselves is this is going to be a risk-based. This is going to be a risk-based approach. We're not going to fall into that trap that so many other people fall into, which is that a risk-based approach really means no risk, absolutely no risk. Yeah, we talk about it as if it was a risk-based approach, but it really means no risk. We were going to stick to our guns and make it a risk-based approach. And what risk is it that we're managing? Well, in our minds, it was two-dimensional. Risk number one was, does it extract at all? The likelihood of leaching, low likelihood or high likelihood. The second dimension was, if something leaches out of a component, what is the likelihood that it's going to persist in the process stream to the very end and get into your drug product? Because obviously things that are cleared can't get into the drug product, don't have a patient impact, and we shouldn't worry. We shouldn't worry about them. So in our mind, this risk evaluation matrix always had two dimensions: the propensity to be leached and the ability to persist. Okay. Again, those of you that remember the history, remember that at one time, 665 actually had a mandatory risk evaluation matrix in it. The risk evaluation matrix that's in 1665 used to be in 665. It was taken out of 665 for the very simple reason that, again, I can count on one hand the number of my industrial colleagues who came up to me and said, boy, I'm so happy that you guys created a risk evaluation matrix so I can throw away the risk evaluation matrix I've been using for all these years that the FDA has seen and has accepted and produces a distribution of risks that at least makes some sense to me. Nobody liked that. Nobody thanked us for that. So we took the risk evaluation out of the mandatory part and put it in the information part. But what we did leave in the 665 was what dimensions we thought the risk assessment assessment matrix should cover. And there were 5 of them. Okay. The material's intrinsic ability to be leached, the process stream's intrinsic ability to be leaching, how strong a leaching agent is it, the driving force for leaching, in other words, the time and the temperature, the elimination of these process equipment-related leachables along the process stream, and the inherent risk associated with the drug product. Those were the 5. Okay, and I have number 1 underlined. Okay, I got all sorts of bad vibes yesterday about how we don't like the way that USP deals with materials. Okay, and I agree. It's an area that's room for improvement. But the simple fact of the matter is if your material is clean and you have little or no extractables, none of these other parameters matter. And the fact of the matter is if your material is dirty and it has a ton of extractables in it, then again, none of these things matter because there's going to be a high likelihood that at least one of these substances is going to end up in the drug product. So for the USP's perspective, this was the most important factor. And from the USP's perspective, that's why we didn't like, frankly, any of the other risk evaluation matrices that we saw out there because no one had this factor in it. And we just thought that was crazy. It was ridiculous. Now, again, in retrospect, I acknowledge the fact that maybe the way that the USP chose to deal with materials is not exactly the most user-friendly way. Again, we lived in the Ivy Tower world that all you had to do was go to a vendor and they would tell you, what was in what was in the material. Obviously, that is the ivy tower. It's not reality. So that becomes a non-starter. Again, this is one of those things that if we knew yesterday what we know today, we probably wouldn't have done. We probably wouldn't have done that. Another approach, that I've seen out there is, well, let's just rank materials. Let's just not arbitrarily. I mean, there's some science based to it. Let's just rank materials: clean materials and dirty materials. I mean, you can imagine what a clean material is from the perspective of organic extractables. Glass is a clean material, and there's other ones as well. And you could imagine what's a dirty material. 33 years at Baxter, a dirty material. DEHP plasticized PVC. Darn it, it's got 40% plasticizer in it. It's a dirty material. Okay, and so polyethylene, polypropylene, polycarbonate falls somewhere in that spectrum. Okay, so that would be a way of putting a material composition into the risk evaluation process. I don't know if we could ever make a concept like that fly, but somehow the risk evaluation process has to account for the intrinsic composition of the material. It just has to, because it's the overriding factor. Nothing can come out if it's not there to begin with, and everything will come out if it is there to begin with. Okay, what else about, so this is my 5 minutes to rag against risk evaluation matrices. It's the mindset that if it involves numbers and it's described by an equation, somehow it's quantitative and somehow it's scientific and somehow it's objective. So if you got a scale of 1 to 9 and then at the end you add them all up with fancy multipliers in front, somehow that's a scientific process. And it's more scientific than if you weigh the individual factors and say to yourself, okay, the more factors that are high risk, the more likely it's high risk. But who's kidding who? I mean, realistically, who's kidding who? The fact of the matter is if you rate contact temperature on a scale of 1 to 9, and 3 is room temperature, and 9 is anything above room temperature, the fact of the matter is the impact of temperature is more than a factor 3. It's as simply as that. A matrix based on those kinds of rankings is inherently skewed. It tends to overestimate low risk and underestimate high risk. Okay? The same thing has to do with the equation that you use. I mean, who says it has to be an addition? Why can't it be a multiplication? Who among us is smart enough to decide between the 5 or 6 factors, which one should be weighted more than the other? The fact of the matter is that the complicated mathematical relationship between temperature and duration and surface area is much more complicated than a simple, we'll multiply this by 0.2 and this one by 0.3 and then we'll add them up. So it's just a process that doesn't reflect the underlying science. So of course it's a process that in some way is going to produce nonsense. It's going to produce nonsense. And at the end, where does the scale come from? Okay, so 1 to 3 is low, 5 to 9 is higher, so on and so forth. Where does that scale? What is the scientific justification? There isn't one. There isn't one. The only way that one validates a risk evaluation matrix is by using it and seeing if it produces nonsense. If it produces nonsense, it's not a good risk evaluation matrix. So I've heard yesterday, and I've heard often. I've been in this business for more years than I care to admit. I've seen the outcomes of risk evaluation matrices. They're either skewed really high, in which case I go back to the client and say, why in the world are you doing this to yourself? This is not the way it should be. Or they're skewed low, and I go back to the client and say, well, I'm sorry, this is just not credible. There has to be some component in your manufacturing process that's higher than low risk. Okay, so what's the good news? The good news in this is that Dennis says, if you don't like the outcome, adjust the matrix. And it's not just my opinion, in quotes, the BPOG, a document right underneath this risk evaluation matrix, says individual companies may use an equal weighting distribution or may assign weighting levels according to their own internal policies. So my advice has always been now to clients and to people in general, if you don't like the outcome of the USP 1665 risk evaluation matrix, and you don't like the outcome of the BPOG matrix, and if the reason that you don't like it is because it just doesn't make sense, you end up with components that when you look at it, you say, this is just wrong. There's no way this can be a low risk component, or there's no way this can be a high risk component. Then my advice to you is to adjust the matrix. Adjust the matrix so that it makes sense. Okay? It's as simple as that. Now that, again, you can't go to the FDA and say, hey, I heard Dennis on March 27, 2024 say, go ahead and adjust the matrix. And that's good enough. And they'll say, oh yeah, Dennis said that. We hate Dennis. By the way, the FDA hates Dennis. Don't ever tell them Dennis told you to do it, because it's the surest way to get a deficiency. But my point is that you need to be prepared to justify. They'll ask you questions about your risk evaluation matrix, and you're going to have to be able to look them in the eye and say, listen, because of the nature of my process, distance along the process stream is the most important. Okay? Other people will make the argument, it's contact time and temperature that's most important, and that's just fine. But you need to be able to explain to a scientific colleague, why that risk evaluation matrix makes sense in your particular situation. And once you're able to do that, the outcome is whatever the outcome is. If 99% of your components end up in low risk, and you can justify why that's happening, that's not a bad scenario. If 99% of your components end up in high risk, though, I've got to tell you, whatever you did to adjust your matrix is not working. Okay. This is going a little bit slower than I thought, but that's okay. We're having fun, aren't we? We talked a little bit about this yesterday, this 85%. Where did this USP strategy of 85% come from? And so let me do an illustration here. And my illustration is my glove illustration. Now you're going to look at these gloves and you're going to say, hey, those are pretty ratty. That's what my wife says all the time. Those are pretty ratty gloves. You look like a homeless. You look like a homeless person. So obviously they're not very aesthetically pleasing. And again, you look at him and say, boy, I hope he's not going on an Arctic excursion in these kind of gloves, because man, his hands are going to freeze. But most of the time, these darn things keep my hands warm. And that was the whole point. The whole point was, if I meet somebody I want to impress, I just put them in my pocket and they don't see that they look so ratty. That's what the that's what the USP said. We're gonna the one size fit all. It's the one size fit all solution to how to qualify manufacturing components. Okay. And I will, again, confession time, bad Dennis for lying to everyone. Yesterday, I, kind of said that the 85% was coincidental. The fact of the matter is, it's actually not. You may remember that at one time there were opposing views on how to risk evaluate. There was the BPOG view and the USP view. We frankly were surprised at the BPOG view of what we consider to be, wow, those are pretty extreme conditions. Why in the world would you do that? So we asked them, how did you come up with those conditions? And they said, we did a survey. We did a survey of all our members, and we asked them what their process conditions are. And we took that universe. And we said, we want to be at the extremes. We're going to design a standard extraction protocol so that no one can think of a manufacturing scenario that would be outside these extremes. And so we thought a little bit and we said to ourselves, well, okay, at least we understand it. No, we don't necessarily agree with it because, man, if I was sitting in the middle and I had a test of these extremes, I'd feel awfully badly used. I'd feel like I was generating awfully exaggerated data when I didn't have to. I'd be justifying extractables that I would never see, certainly not ever see at these levels. So I thought, the USP thought, boy, that would be very uncomfortable with most of the people. So we took the other approach. We took the same data and just said to ourselves, hey, we want to be in the middle. We want to cover a majority of the operating conditions out there, a majority of the most common operating conditions out there. And it turned out that was pH 3, pH 10, and 50% ethanol. And when we looked at the data that users had supplied in terms of what their process conditions were, that was 85%. Okay, so we felt 85% of the people were going to look at the USP extraction conditions and say, it fits. Maybe not very well, but it fits. Okay, these are them. We talked about NVR and UV absorbance yesterday. There's room for improvement. I'll admit it. Everyone is asked for a specification, pass or fail. A revision of 665 should contain pass or fail criteria, but they're going to have to be customized. In the same way that we customized extraction temperature as a function of the individual components, we're going to have to customize the NVR and the UV requirements, because an NVR and UV requirement for tubing is not going to be the same as for a filter, as for a bag. So it's going to take a while to accomplish that. But again, the industry has spoken. They want acceptance criteria, and you'll get acceptance criteria. You won't like the acceptance criteria. Nobody ever does. And again, I'm going through this really quickly. This is what's in the chapter. I guarantee you it's in the chapter. You can easily read this. I don't need to talk to this. Okay, we talked about NVR needs improvement. Okay, yeah, here's a good subject. Let's talk a little bit about what 665 says about leachables testing. Actually, 665 doesn't say much at all about leachables testing. Oh my gosh, did the people who wrote 665 forget about leachables testing? Did they forget that it's E&L and they only did E? No, we didn't. We didn't forget. But yeah, let's go there now. Let's go there now. Let's go there now. We might as well go there now. Okay. Again, please don't go to the FDA and say Dennis said this, but I'm going to say it anyway. It's much to do about nothing. The USP believed from day one that this whole issue of extractables and leachables was just an overblown issue, and that if people could just think rationally they would understand that the actual risk that leachables from manufacturing equipment would end up in the drug product at levels high enough to cause a safety concern was really, really little risk. It was a really small risk, and yet we were requiring all this testing. But understand, it's the fact that we required the testing that produced the data that people could use to make good decisions, that get us to the point now where, again, I'm going to justify to you why the USP was right when they say that the risk of leachables, manufacturing-related leachables, getting into a drug product at levels that are unsafe is vanishingly small. One, again, in our mind, the most important reason was vendors played the role that they were supposed to play, and users played the role that they were supposed to play, which is, I'll be damned if I'm going to use a component that's dirty. Okay? BPOG stood up and said that. We'll be damned if we're going to use components that are dirty. So you're going to have to give us data to prove that the components are clean. And the vendors do the right thing by saying, yeah, we hear you. We are going to develop components that are intrinsically low in extractables. That's the way it's supposed to work, and that's the way that it did work. So most of the components manufactured today by reputable manufacturers are low in extractables to start out with. Okay? And we've always known as a community that the driving force for extraction for manufacturing components is not as aggressive as it is for a medical device or as it is for a container closure system. So the ability to be leached is less in manufacturing situations than it is for the other two. When we first got involved with this, clearance was a theory. There were people out there, and I'm looking right at one of them, who knew it wasn't a theory in 2015, but to the rest of the world, it was just a good theory. Here in 2024, it's a verified fact. Clearance happens. If you want to have a debate with me about whether clearance is a reality or not, let's have that debate. I'm going to pull out the 10 or 15 or 20, however many publications there are now, and prove to you that it's a reality. And if clearance is, if your materials are clean to begin with, and the leaching ability is small, and oh, by the way, whatever does extract is cleared, wow, that's a pretty good scenario to be in. The other point, and again, this doesn't show up in risk evaluation matrices very often, is it depends upon what dosage form you produce. I mean, is it an oral dosage form? Well, gosh, I've got it on record where the FDA says for oral dosage forms, container closure systems don't even need extractables or leachables information. So why am I doing extractable studies on manufacturing components to produce oral dosage forms? And what about a vaccine? What about a vaccine that, maybe you inject a mil every year or something like that? Surely we can accept the fact that the risk, that the patient exposure, because the volume and the frequency is so small, is inherently a low risk scenario. And again, point number 5, I'm going to stand in front of you and say that as a client who's seen an awful lot of leachable study, I've never, that's zero, absolute zero, never seen a leachable, a manufacturing-related leachable, reported in a drug product anywhere near a concentration that it could consider to be unsafe. So to me, that's proof that the system has worked. Okay? Now, does that mean we're going to throw 665 in the garbage? There were times, and I'll admit, I've told people that are in this room that I thought that 665 was going to go away. that there would be a point in time that we would collectively realize that the risk was so small that it would make more sense to just have somebody, who that somebody is, I don't know, say, hey, I've seen enough you'll know who I'm talking about when I say this. We've seen enough submissions that use this component that, I don't want to see another submission that uses this component. I'm just going to say it's safe. You use this component, it's safe, it's fine. Why can't we get to that? Why can't we get to that spot? But again, the way you get there, you got to do the testing. You got to get to the point where you have the data that justifies that conclusion. So the simple fact of the matter is 665 isn't going to go away. People are still going to have to generate 665 data because it's the means to get from point A to point B. All right? One last thing, and I know, Carson, I'm way over time, and I'm apologize for that, but one last thing that I want to point out. The thing that, this never even occurred to me until I began to see it in tox assessments. As an analytical chemist, the number one item that pisses me off is when I spend hours and hours and hours and hours elucidating a structure and getting the concentration exactly right. And the tox assessor says, well, I didn't even need to tox assess that compound. And you say to yourself, wait, but it's above the AET. Aren't you supposed to tox assess everything above the AET? And what they tell me in manufacturing is, yeah, but when you calculated the AET, you forgot the fact that the manufacturing production process produces more than one dose. The extractables that come from that filter don't end up in one vial. They end up in 1,000 vials or 10,000 vials. And so all of a sudden, you end up in a scenario where, yeah, I quantified and identified down to the AET, but the tox assessor takes into account, well, the 10,000 vials were produced. So they're not going to tox assess anything that's below 10,000 times the AET. So I strongly, I'm not suggesting that you necessarily raise the AET to take into account, because now the AETs are so ridiculously high that there's no peaks above the AET. But you remember that when you have an unknown that's right at the baseline that nobody has a clue on how to identify, maybe that's an unknown that you can just let slide. Anyway, with that thought, thank you very much.